DNA sequence? Well, this is the heart of what we're doing here. It's what distinguishes us from one another and from other animals, our DNA sequence. It's just a string of bases, given these symbols, A, G, T, and C, as I mentioned, and it's the sequence of those that determines everything about you. There are two types of DNA. The nuclear DNA in the nucleus of the cell is from both of your parents. You have paired chromosomes. There's 23 of them, and pairs, and one from each parent. Whereas the mitochondrial DNA is a much smaller molecule, only 16,568 of these bases in a circle. And this you inherit strictly from your mother. So the line goes from grandmother to the mother to daughter, or any siblings. All siblings will have the same DNA, but only the girls can pass it on to the next generation. So that's a big difference here. And yeah, this circular DNA down in the bottom there, it comes from the mitochondria in the cells, which are related to the energy processes, storage, and formation. This is all cell biology. The part of this circular molecule of 16,000 plus bases that I'm going to deal with is just a small part. It's called the 12S rRNA. We don't need to go why it's called that, but it's a small segment, 220 some bases only out of 16,000. But it's enough to distinguish different species, fortunately. Okay, so that's DNA. What is environmental DNA? Well, as you guess from the name, it's what we find in the environment. At first, we don't know what's in it. It can come from soil. You can take it off the footprint or any other soil sample. Air has some, but only in fine drops of mist. So pure air will not have any DNA. It's not volatile. But if it's in drops, it could be. Somebody sneezes, for example. Water is a great source. That's what I'm going to talk about, collecting water samples up here. And you get the DNA, as I mentioned before we got started, from the fish, a lot of them. Salamanders, reptiles, turtles, any snakes that go through there. I got one from a water moccasin. Mammals, they go to drink. They relieve themselves in the woods. It washes into the water. So you get just a plethora of things, and the job is to focus on that and figure out what could be Bigfoot. And so ice is another one. If you found footprints that look like Bigfoot, collect the ice from the footprint, let it melt, and run it through the filters that I'm going to talk about. And it's just like a water sample then. And you might find some Bigfoot DNA in ice. And any other surface, people have collected it from trees to see what kind of squirrels and stuff. You know, the DNA gets sloughed off of the skin on the bark. So it's very, very generally applicable to wildlife studies. Some of the examples that have been successful, Okinawa, that island owned by the Japanese, was the last major battle of World War II. Some folks analyzed the DNA in some of the ditches looking for fish and so on, and by God, they found salmon, which are not native at all to Okinawa. But the ditches have waste streams. The septic systems aren't that great. And so, because the people ate the salmon, they love salmon in Okinawa. You probably had you know, various Japanese dishes with salmon. And, you know, they excrete it and it ends up in a ditch. Reef fish, it's hard to go down. I'm not a diver, but I would imagine to do a survey of fish on a reef would be quite a job, and you'd never be sure you saw them all. But if you collect a water sample, do the DNA analysis, you'll know what different fish are there, if you've got some reference sequences to compare from an aquarium. And then, they found some mammals and pond water in Japan. That gave me the idea that I could do this in the streams of the Ouachitas. The Loch Ness Monster, have you all heard about this? Neil Gemmell from New Zealand took a crew up there and they collected 250 water samples at different places and different depths in Loch Ness. You know about the Loch Ness Monster, right? Well, that's what they were trying to find. And if they had found DNA that was something different, they might have found it. However, they found 3,000 species, including microbes, in all the birds and the animals that ever got anywhere near it. And they did not find anything that looked like it could be a Loch Ness Monster, except they found a lot of eel DNA. And eels, like a lot of other fish, some of them, they have no limit to the size. It's only when they die that determines how big they get. And there are some eels, the European eel is only about 30 inches long, but some have reached several feet. And so that's a possibility. It's not proven. It's one of those things that he's on the right track, but more work needs to be done. Now, aquatic, I've already said, aquatic DNA can cover a huge area. You can potentially cover a whole watershed if you're in the right place and it all washes in. So that's good. We have two approaches. We either pump the water out with a peristaltic pump, force it through a filter, or we use a syringe to draw it out and force it through a filter. And it's the cells that get sloughed off. Now, fish, of course, they're exuding DNA like crazy, and other animals, not quite as much, but enough. Then you either freeze it, or you add a preservative, and then you can mail it to the sequencing lab. All right, here we are. Now, somebody like David Wilbanks can probably tell where this is from his experience. But this is in the Ouachitas. I took water from several creek samples, a river, guess what river, the Kiamichi, and then some ponds. The ponds are very interesting, we'll talk about later. The first sampling technique for one of the labs I used, you see here a Stereovex filter in the center of the picture there. And of course you wear gloves and you hook this up to a pump. You run it with a battery-powered drill with a very large bit that fits this little slot. Yeah. And when you run the drill, it runs the pump. And so the water goes in from the left there, and to the filter would be on the right. The setup in the back of my truck, you can collect your sample in a bucket and then at your leisure filter it, or you can collect it directly from the stream or body of water. So that's the setup there. It's pretty simple. The water sample is below the pump, goes through the pump, and then the filter's on the right, and the filtrate, the water is just discarded. Then you put it in the cooler if you've got that kind of filter. Keep it cold till you mail it. Then you got to mail it frozen, and it costs a lot of money to do it that way. It's much better to do this other approach in which they send you a kit, and in it are the things on the right. Starting from the left of the right-hand picture, there's the syringe, 60 milliliter, some gloves. You always wear gloves. You don't want to contaminate it with your own DNA, but I am very lucky because my DNA in this 220 base region is exactly the same as the human reference sequence that we're going to compare things to as a standard. So you're all comparing to the same thing. And so if I contaminate it, I won't even know. It's just, there's no extra mutations. That's great. Then you have on the right there, the right-hand picture, the filter. It's a disc, and it's in a little bag, and it's barcoded on the back, or Q-scan, and so you keep track of it that way. There's some caps to put on it afterward, and a little syringe of preservative. So the procedure, I could have brought one, but you got a good imagination, you people. You wouldn't be here. You suck up water. You clean your syringe. You do that 3 times. Then you suck up your actual sample. You put the filter on. You can't put the filter on first because it's a one-way kind of thing. It only goes out. You cannot suck up through the filter. So you got to put the filter on after. You squeeze the water out. That can be discarded. And you keep doing this until the filter is so plugged that you can't squeeze it anymore. So it might be 4 to 6 or more. The more the better. The more DNA you get, the better. And then you quit. You throw your syringe away. Well, first you blow some air through to get the extra water out. And then you take that little syringe and inject the preservative. Put the 2 caps on the end. Put it in the bag with the barcode. Record the barcode and where you got the sample and all that stuff. So you know which is which, and send it off. And what they do, they'll break this down, breaking down the cells and extracting the DNA, and then breaking the DNA into smaller segments, amplifying it. There's a process called polymerization chain reaction, and there's not enough DNA in here to analyze directly. So it has to be replicated. And there's a really nice instrument to do that replicates more than 1 billion times the amount that you have. Just copies each strand. I won't get into all the details, but that's important to get enough sensitivity. And then they do the sequencing part. There's a couple of ways to do it: by adding bases and noting what that base is, by fluorescence tagging, used to be radioactivity, or something like that. And then they will report a sequence. I always demand to see the original sequence, not just a list of what they found, because I find stuff they didn't find, and I get extra data that way. So they'll report the species that they found, and you can get the sequences and check it out yourself. Now this is a little video. I'm going to skip that because it's really hard to get it connected. You get a file called a FASTQ file for each little cluster of that sample. There's many, many, many millions of them sometimes. And each one has a sequence in black. And also error codes. This is important. Those error codes can be translated with some simple formulas into the probability that that sequence call is correct. And you need to know that. We like to make it 99+% correct. And if not, then we've got a problem. So you analyze and you take these sequences and you search the database. The database is called GenBank. It's in the National Institutes of Health, the Biotechnology Center, and you search it with online BLAST It's called software. It's a search engine. And people that do this kind of research submit their sequences to NIH. And they put them in this database for other people to see if they found the same thing or not. Human and all the other plants and animals that you can think of that they've studied. So this thing has millions of species by now. And it's a moving target because every day somebody submits something. I submitted some samples, as a matter of fact. And then you will search engine, both NIH, free. One thing the government gave you back for your taxes that most people can't use, unfortunately. You get a result from this search. This particular sequence, you see two sequences in each row here. One is the database called the subject, and the query is your sample. And where the lines are vertical, they match. And if there's any gaps, they don't. This is 100% match with no gaps to, anybody know what that species is? Odiocoleus virginianus. White-tailed deer. There must be some hunters in the audience here. So we get deer. They come and drink the water. Okay? I found about 19 mammals, I think. That includes myself or other humans. It's quite a variety of what you'll find here. This is just in the Ouachitas. There are some domestic animals. There are some others. I was surprised to find the beaver. I thought they were extinct east of the Mississippi, but not true. They're here in Oklahoma. And foxes, coyotes, possums, squirrels, rabbits, even a bat. Okay, the red bat probably flies over these streams eating bugs. And as it does so, just like birds, they poop at times in the water. Birds, not as many, but those two on the lower right, those are little birds. The red-eyed vireo and the hooded warbler. The red-eyed vireo, I found out, nests near water. So that makes a lot of sense. They're pooping in the water as they go to the nest or the babies. And, you know, just a little bird like that, you find its DNA. So that encourages me that maybe I'm onto something. Fish, a ton of fish, of course. I mean, this is ideally suited to fish because they're in the water all the time. They're sloughing off from their slime and from their mouth and God knows, and their poop. Amphibians and reptiles are kind of interesting. I found the Ouachita dusky salamander, which is a very endemic species here. That's the picture of it. I was really happy to find that. I found other salamanders as well. Okay, getting to the human side. Once you've done this and you kind of know what's out there, you're finding some human, or I'm going to say human-like DNA. And in order to compare these all on the same basis, there's a reference sequence in the database that was produced by the inventor of the first sequencing of DNA. Frederick Sanger won the Nobel Prize twice for sequencing proteins and DNA. And he had a woman's DNA that has become the standard. You can't just compare randomly because then people coming in to look at it, they have a different reference tape. But if everybody has the same human reference, then we can make good comparisons. And here we are comparing over this base range to me, and it matches 100% here. So if I pollute the sample, I'm not going to see any different mutations from the reference because I match it perfectly. That's very fortunate. One of my colleagues did contaminate a sample, but we had his DNA, and so we simply excluded that mutation. And you get your DNA, you know these companies, Family Tree DNA, and it's pretty cheap. I advise you, if you're going to get into this, You need to know, and anybody who works with you needs to know what their full mitochondrial sequence is. And then you can exclude, you know, any kind of contaminant if it occurs. Now this is where it gets real tough. Sequencers have natural errors. All instruments do. I don't know of any that are perfect. How am I going to distinguish sequencing errors from real mutations? That was the crux of this whole thing. And what I do, on the bottom, each base can do 5 things if it's going to do any changes at all. It can mutate to one of the 3 other bases. It could be deleted, or there could be an insertion there. These are the 5 possible errors. So I've plotted them along the horizontal axis for just a small segment here, 883 to 952 on the reference system. And I've plotted the percentage of these in each sample. Each sample is a different color up there. And notice, this is amazing. There are spikes in just certain places. These are where the greatest number of sequencing errors occur. They stand out. But in those are some bars that I've pointed to them with arrows. You can't see them, but trust me, there's some of the bars in there from some samples that are way bigger than others. These are the ones that are the real mutations. Okay, they stand up above the sequencing errors. Here's an example. Here's a sample, SD1, and it's over a short range here, 1022 to 1090. And here are the percents of total mutations and errors. And notice at position 1048, there's a huge spike. This is not a sequencing error. The sequencing errors are those little things you can barely see on the bottom. And this one stands up so much above, it is a real mutation. And it's an interesting one we'll get to. Now, how do I know, comparing one sample to a bunch of others that were collected at the same time, looking at a single base position, that my sample of interest is a real mutation as opposed to all the others which have a sequencing error at that position? I use statistics. Melba Ketchum said this about me. He uses statistics. I don't know if that was a compliment or more than likely she thought it was not a compliment. But anyway, there are tests. If I give you these numbers, 8, 10, 12, 13, 14, 15, 20, as measurements of supposedly the same thing. You would look and you'd say, well, they all go up to 15, but then there's this gap and there's this 20. Is it a real outlier or not? And there's a statistical test, Grubbs test, or Teach and Moore if you have more than one, that can tell you with what probability this 20 sitting out here above the 8, 10, 12, 11, 13 is an outlier or not, and at what probability it would be. And so that's what I use. Otherwise, it's a matter of, is this bar bigger than that bar? You know, and that one, and that one, and that one. Is it bigger, and how much, and is it significant? You know, you gotta use statistics to tell that. Well, here's some comparisons. People make a lot of comments that human and chimpanzee are 99.9% true, or 99, they'll say sometimes. Actually, they're not that close, especially in the region that I looked at. But here we have up above that gray bar is the reference sequence, and over this region, Neanderthal differs by that one red dot over here. That's a mutation. Gorilla, chimpanzee, and orangutan by much more. there are many more red dots where there's a difference. And so that's the point here. Here it is from the output of the blast search. Top row would be the reference sample, bottom row adjacent to it, the Neanderthal. And there's this one place where there's a gap and they don't match up. Position G1048 is mutated to an A in the Neanderthal. So we're pretty close to Neanderthal, right? You knew that. But chimpanzee, you know, that's not 99.9%. That's about 96% the same. The red dots are where we differ from the human reference from the chimpanzee. And that's important here. Gorilla, similar, even worse. Orangutan, even less of a match, more red dots. So it should be easy to distinguish a human or human-like species from apes. And that's what I'm hoping to do. Oh boy, this gets a little bit technical. Haplogroups. In the whole human population, there are about 5,500 known distinct mitochondrial DNA differences. That's the circular one with 16,568 bases. So we all fall into one of those, and occasionally we have a few extra, very few extra mutations. I have 4 extra ones. I'm H22 with 4 extras. And what do these symbols mean? Online, there's a great site, phylotree.org. It gives you all these human DNA combinations for mitochondrial DNA. And they're all organized. It's like a tree. It starts with mitochondrial Eve. She's the one that we're all related to some 100 to 200,000 years ago. And all the DNA from there is represented in this tree of mutations. And this is just the top part of it. You've got to dig deeper by clicking on one of those little squares at the bottom, and then you get to see the fine details. Like, for example, that mutation that I pointed out that stuck up, it's found in 2 places, D5B, there you see it circled, and M29. Well, where do these come from? You know, if this is a Bigfoot sample, it's obviously human-like. It's got this human mutation, but where would that be from? D5B is totally Asian, all over Asia. It's a common one. M29, this gets me back to my World War II history, is found in the Solomon Islands. That was the first landing that we made in the Pacific War, Guadalcanal, you may remember. So that's a rare one, but D5B is not so rare. And this shows just the migration patterns, these haplogroups, these combinations of mutations. They've got them traced to how they all came out of Africa. All right. That D5B, I looked up, one nice thing about this database, GenBank, is you have a full sheet of all the information on each sample. Who collected it, the reference, the literature, the authors, sometimes the haplogroup, and other information, plus the sequence. And these are the countries that it listed for D5B. So it's fully Asian. It's interesting how you know, it covers almost the whole part of Asia. And this one, the Solomon Islands, much more limited. Very interesting. Now, the bottom line, well, how does this Solomon Islands relate to the Ouachita Mountains? Well, these are the mutations inside each little box that I found in the various locations. I tried to put them in the map where they occur. And these are the ones that have more than one instance. So they're not just an unusual thing. And also I had a colleague in Alabama who took some samples, and her results are over here. Now, I drew a few arrows to show that there's more than one occurrence, like that far upper left, 918G. Well, it occurs 978G. It occurs over here in the middle too, and then down below on the right. So I didn't draw all arrows, it'd be too confusing, but the point is there are multiple occurrences of rare mutations that in the database are either not found at all or rarely found. And so my conclusion here is that these could be human, but they're more often likely to be human-like because a lot of them aren't found in the database. Not all, but a lot of them. And because of I don't know, since these are rare, would a person be, this is several miles in each direction, up and down this area. And a hunter, I don't know if he'd cover all that area and be found way over here and then over here and over there. I don't think so. I think it's a more mobile species. Okay, my conclusions, and please don't go home and say Haskell Hart thinks he found Bigfoot, because these are tentative. Numerous vertebrate species, no uniquely ape-like sequences were found. I first checked each of these samples to see against the apes, and there was no match. There was one match that was similar to human. So it turned out that it was a nuclear incorporation of this mitochondrial, and that it happened way back before the gorilla and the human split. But it wasn't uniquely ape. Unusual, rare, and non-human mutations were found in otherwise human-like samples. That is, the rest of the sequence was human. 5 of the 6 ones found in Alabama were also found here in the Ouachitas in Texas, in Oklahoma. So that's kind of unusual. And finally, number 4, this I really want you to remember this one. Evidence is consistent with, but does not prove, the existence of a human-like primate, very distant from apes. This is what David Wilbanks hypothesizes too. And, it's evidence. It's like footprints. It's like howling. It's like wood knocks. What else do we have? Rock clamping. Yeah. Rock throwing. Very good. It doesn't prove it, okay, but it's close. And then you have all of these together, then you're getting closer to proof. I still think, like all of you would, that we'd like a body or a good part of one. But this is evidence. It's not proof. Well, what I'd like to do in the future, I'm getting old. I'm 82. I need to pass this on. Form a consortium where people collect these samples in different places. And they're not expensive. I'll tell you, there's a setup fee of $150, and then it's $105 a sample. And they won't take anything less than about 6 samples. So for 6 samples, it's $630 plus $150. It's $780 to do 6 samples, which is not that bad for the information. And of course, we'd like to find a body part. Now there's 2 new developments that I'm going to tell you about here for the first time that I've used. Most of the rest of this I talked about last fall in October. Have you all heard of LIDAR, which is light detection and ranging? It uses pulsed LIDARs from an airplane to go down, and it goes right through trees and shrubs and gives you the topography of the landscape. You've probably heard of these cases in the Mayan jungle where they found huge cities that were covered with trees and forests. They grow up, I've been to one in Belize, they grow up right through the pyramid trees. You can't see them. You have to either explore it on foot, or LIDAR brings them out beautifully. I should have put one in here. Well, I got a staying closer to home. Garden Ridge is like 15 miles from where I live. And there's a famous bat cave there called Bracken Bat Cave. And here is the LiDAR signature that on the left, I've drawn in contour lines. I didn't draw them, the computer did. I have software called Global Mapper, and it's from a place in Maine. And look at that on the left. Well, you can't tell if that's a hill or a crater, actually. But when you look at it in detail and look at the numbers on each of these contours, you'll see that that is a huge cave. And I think we should be using this. We need to find out where these things shelter when the weather gets bad. But I used it. This is on the Kiamichi Mountains. And I'm sorry this doesn't show up better. There's too much light. But in the middle of each circle, there's a crater-like. And it looks, there it is. This little thing, I thought they were meteor craters. But they're all perfectly round. There's none that seem to come in from an angle. Something wasn't quite right. Now this, that's a blow up. But this is what you, on a topo map, you wouldn't see this. The elevation of this crater is only a couple feet above ground. And so the little red circle is where it would have been. That's a topo map. It wouldn't show. How about Google Earth? Well, there's a little thing here. I'm sorry, there's so much light or something that just doesn't show it. There's a little evidence of something that goes below the trees there. This is what it looks like. When you use this software, you can get the exact coordinates of that crater. And so I did it. I went there. It's on Forest Road 6025. It's only about 40 yards off the road. But you'd never see it. And all that undergrowth and trees. And the best time to go is in the winter, actually. I went at the end of the winter in March. And I was able to see it from the road. And I also figured out where I'd have to park my car and at what direction would I have to walk to get to it. All that is on Global Mapper. It's a piece of cake, really. So that's the little pond, and my thoughts were that maybe Bigfoot uses these as a water source. They're up on the ridge. There's no other water sources, really, permanent. And so that's one. Here's another one. They're yucky looking, and they're full of algae and bacteria and probably salamander eggs and all kinds of stuff. You don't get much with a couple of syringes. It plugs pretty quickly. All right, finally, it's not exactly my invention. I picked up on some other things. Detecting humans or human remains at a distance, even if they're underground. Okay, here's how it works. On the left is a source. It can be your cell phone, but it emits a certain frequency. Now, it's in the audible range, but it's the electromagnetic component that we're using here. Because what is a speaker? It's a little magnet that goes back and forth due to electric current, and so you generate electromagnetic waves of the same frequency as the sound. And so that goes along, and there's your source on the right. It can be a human, live. It can be bones, a skull, a dead human skeleton, a tooth. But all of these have in common DNA. And I calculated with that frequency, would it be the right frequency for a proton to go around this circular DNA, and it comes out pretty close. So I don't know for sure if it's the DNA, but all these things have that in common. They also have collagen, which is a protein, and that might be a factor. I need to do more on that. So what you do, you set your source up. You have aluminum rods. Now you're going to think dowsing and all that, but those rods are antenna because they're made out of metal. And you hold them, they're stuck, they're bent, They go in a PVC pipe for you to hold it without touching the rod. And you go like this. And when you cross the line between the source and your target, which you don't know where it is, but when you cross that line, the two rods go together. This is the same phenomenon that people that deal with underground cables use to find them. All right? They do the same thing. And it's still the electromagnetic wave that causes that interaction. And so you go around. We'll say rod is the target. And I'll get an alignment. Okay, so I have a line now. But I don't know how far off it is. I keep going. I try again. I hit that same line. And then when I get close to him, we have another very good phenomenon that tells me exactly where he is. That's walking the rods. These hands are my hand, the blue are the rods. You see where they line up along the line between the source and the target. But when I get close, and I did the calculus on this, this is a true thing. When you get close, no matter what direction you come from, there's a circle in which the rods align tangentially. And not only that, the diameter of that circle, if you take half of it, the radius of that circle, which is D, I'll call, is equal to the depth of the target. How convenient, how nice. And this is proven. I did it experimentally, I did the math. It comes out exactly like that. So when you find this circle, take half of the diameter, and that's your depth, right in the center. Now this is not magic. It's not supernatural. I'm sure you all remember Maxwell's equations from vector calculus, right? These equations were the ingenious development of a 19th century physicist. These are over 100 years old. They relate electric to magnetic fields, especially when they're changing. And every time you have a changing electric field, like on a wire, you're going to have a corresponding changing magnetic field. Every time you generate a corresponding magnetic field that's spinning a magnet, you're going to generate a current. The two are inseparable. And the equations that are of interest is delta B is the rate of change of the magnetic field produces an electric field whose curl function is that. Now, you don't need these details. The point is the arrows. B produces E, or E produces B. And that's what's happening. These little rods, when I go, I'm changing the magnetic field, so I'm causing the currents in the rods. And those currents generate their own magnetic field, and those two little magnetic fields act like magnets, and they draw these two rods together. Okay? That's how it works. It's not the peach sticks and the water, okay, the defining rods. I'm sure I don't know how that works, but I do know how this works. And so I've been using this. You go to a cemetery. I found graves of children that died of some horrible disease over 100 years before. I've seen a guy who was getting marijuana from a field from over 100 yards away. I detected him. And I detected the marijuana with a different frequency. So you can change the frequency for different things. And so I did not invent this. Somebody told me about it. And apparently Raytheon says they already know it. So no luck on that. Well, I want to thank Susan Barron. She helped me collect samples in Alabama, Kim Gerhard in Oklahoma here, not too far away, the Ouachitas. And the funding, I funded the Oklahoma samples, and Susan and I co-funded the Alabama samples. I've spent a lot of money on this, but I'm happy to do so. Now, if we had an internet connection, I'd be playing you this music. It's from the Coasters, 1957. I'm going to find her. I'm going to find her. Da, 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 da. I'm going to find her. I'm going to find her. Yeah, I've been a-searching. Da, 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 da. Anyway, could we do an internet connection? Or is that, where's Brian? It's a great song that I didn't discover until 60 years later. A little slow on that one. I wasn't real cool at that time. Brian, can I get an internet connection? Let's just click on that. You need to hear this song. Oh. Well, I don't know. Do they have a Wi-Fi here? Yes. If they have Wi-Fi, you should be able to click on that link. Let's just see what happens.
Well, first thing I'll have to do is I'll find, where's my, oh. Let me minimize that. Minimize that. Oh, wait a minute.
I just clicked on the...
There we go. And it looks like we're connected.
Okay, can you click on that one? It's the last one.
Yes, we're connected. Hold on.
And then you get to the site and you got to click on another button.
Why is it? Oh, okay. That one?
The blue one. Yeah, that's the link. I just clicked on it at home on the computer. There's the words down there. Oh, yeah, yeah.
Is this at the bottom?
Right here, this blue link. Yeah, it's at the bottom. On the last slide.
Okay.
Last slide. Keep going. Couple more. One more.
I swear my computer works fine in my office.
Well,
there it is.
click on it and see what happens. Oh. Good grief, we're getting your words down below. Oh, well, you can imagine. Look it up. It's free online to hear the coasters.
Oh, that's weird. I'm actually changing here.
Just left click, right click, right click.
Font. Yeah, I was.
That didn't work? Okay.
Kind of sounds like the military. You're doing your list.
Oh, really?
Open link.
Open link. Okay. Now, here we go. Turn up the volume. It's a she, just like Patty.
You need to dance before it gets over.
I wish I could. When all my colleagues were dancing to this, I was studying calculus. That's how you got the Maxwell equations. Aren't you glad? Anyway, that's it. Thank you, folks.
Thank you, sir.